10, muscle tissue. So we begin with an overview and an introduction to the chapter and what you will learn. In this chapter, you will learn about the structure and function of the 3 types of muscular tissue. You will examine the events at the neuromuscular junction. You will describe energy use in muscle cells, and you will understand how muscle tension is controlled. We have 3 types of muscular tissue: skeletal muscle, cardiac muscle, and smooth muscle. You should be able to compare and contrast each type of muscle to one another, as well as know the basic properties. Skeletal muscle will be located in the skeleton, and its function is to produce movement or help with movement, heat, and posture. It is striated and has parallel fibers, and it is under voluntary control. Cardiac muscle is found only in the heart wall. Its function will be to pump blood. It is also striated like skeletal muscle, but it has one central nucleus. It is involuntary in control in that autorhythmic fibers, hormones, and neurotransmitters control it, so we don't actually have to tell our heart to pump. Smooth muscle will be in our gastrointestinal tract, uterus, eye, blood vessels. Its function is peristalsis, a special type of movement, and it does not have striations, and it has a central nucleus as well. It is also under involuntary control, like cardiac muscle. So here's a lovely chart for you that gives you some of the basic functions, but most importantly, so what are the functions of muscular tissue? To produce body movements, to stabilize body positions, so think about our posture, to store and mobilize substances within the body, and to help generate heat, or the process of thermogenesis. Muscular tissue has a few properties. It is electrically excitable. We had one other tissue in our 4 main tissue types that was electrically excitable. Please make sure that you remember that. It has contractility, extensibility, and elasticity. This will allow your muscle to contract and then actually return back, and it also allows it to stretch. Now, let's go over the connective tissue components that make up our skeletal muscle. On the next slide, you'll see a picture of these, but here we've listed them so that you have them all in place. We have the subcutaneous layer, the endomysium, perimysium, and epimysium. The endomysium will be the innermost layer, perimysium the middle, and the epimysium the outer layer. Then you also have the fascia, which is a sheet or a band of fibrous connective tissue that's deep to the skin, and it will surround muscles and other organs of the body. If the connective tissue layers extend beyond the muscle to form what we call a rope-like structure, it's a tendon. And if it forms a flat sheet instead of the rope-like structure, it is called an aponeurosis. And so fibromyalgia is a disease process that affects this connective tissue layer of our skeletal muscle. So here we're just simply looking at a picture of the skeletal muscle, a transverse section. And we have the tendon connecting the muscle to the bone. So remember that tendon is a rope-like cord. That tendon will then be continuous here with the epimysium. And then we have the perimysium, which is going to surround an actual fascicle, which is a group of muscle fibers. And then in the actual fascicle, we see it blown up with the perimysium surrounding it. And then we can see the actual muscle fiber cell. Here on the muscle fiber cell, we then see the endomysium. And then we can see each individual filament here, which we'll get into details about what's contained in the myofibril, or all of the filaments that make up the myofibril. So when we are looking at these filaments, we have to do so on a microscopic level. And so the main microscopic anatomical parts of the skeletal muscle fiber are the sarcolemma, the transverse tubule, also known as the T-tubule, and the sarcoplasm. These T-tubules are tiny little invaginations of the sarcolemma that quickly spread the muscle action potential to all parts of the muscle fiber. So that's why we have them. And the sarcoplasm would just be equivalent to a cell's cytoplasm. So sarcoplasm because it's in a muscle fiber. And then we have a triad here that is made up of the T-tubule and the terminal cyst on either side of it. So the terminal cisterna is what we call the dilated end of the sarcoplasmic reticulum. So just like we have an endoplasmic reticulum in cells, in muscle cells, we have a sarcoplasmic reticulum. And so we have a terminal cisternae at the dilated end of those that will allow, and so on either side of the T-tubule, we have that, and we call that the triad. The sarcoplasmic reticulum will encircle each myofibril. It is similar to smooth endoplasmic reticulum in non-muscle cells, and in the relaxed muscle, it will store calcium ions. When we release calcium ions from the terminal cisternae, it triggers our muscle contraction. The sarcoplasm is the muscle cell cytoplasm and contains a large amount of glycogen for energy production and myoglobin for oxygen storage. So the sarcoplasm contains glycogen and myoglobin. Next, we have our filaments and the sarcomere. The sarcomere is going to be the individual unit of filaments. And so we have an A band, Z disc, M line, H band, and I band and H zone. So I band and H zone. So here we're showing them. The darker middle portion is what we call the A band. It is mainly made up of thick filaments with some thin filaments that overlap the thick ones. The lighter sides of the I band consist of only thin filaments. Your Z disc will pass through the center of the I band. So the Z disc passes through the center of the thin filaments. Exercise can result in a torn sarcolemma, damaged myofibrils, and disrupted Z disc. The narrow H zone that we have in the center of each A band contains thick but no thin filaments. So remember, we said this A band here contains our thick filaments, but it also overlaps with our thin filaments. So the H zone is only going to be the thick filaments. And then finally we have the M line formed by myomesum in the middle. So our sarcomere runs from Z disc to Z disc is the unit of the sarcomere. And you can see the thin filaments contain actin. The thick filaments contain myosin. We will talk about these proteins in just a few. You also have a nice table that goes over the details. All right, so I mentioned the thick and the thin filaments contain our proteins of myosin and actin, with actin in your thin filament and myosin in your thick filament. Myosin is a thicker word than actin, so myosin will be in the thick filaments. Myosin and actin are both what we consider to be contractile proteins, meaning that they generate force during contraction. Myosin in the thin filaments will be the binding site, actin in the thin filaments will be the binding site for the myosin, and myosin in the thick filament is actually going to be the motor protein. It contains two heads here, and these two heads contain an actin binding site and an ATP binding site. So the thin filament you see here, this thin filament of actin has a myosin binding site. Right now it is covered by one of the regulatory proteins, tropomyosin. Tropomyosin is present in thin filaments. In relaxed muscle, this tropomyosin will be held in place by troponin, the other regulatory protein. And tropomyosin blocks the myosin binding sites on actin, preventing them from binding together. We also have some structural proteins that will provide alignment, elasticity, and extensibility to our muscle. We have titin. Titin is from the Z disc to the M line of the filament, and it will stabilize thick filaments. It helps the sarcomere return to its resting length after a muscle has been contracted or has been stretched. Nebulin will help maintain the alignment of the thin filaments in non-elastic in the sarcomere, and it will also anchor the thin filaments to the Z, to our Z disc, and it helps to regulate the length of our muscle. We also have alpha actin in the Z disc, and this will help bind actin to titin. Myomesum will form the M line, we said, and it binds to titin, and it connects the thick filaments. Finally, the last structural protein is dystrophin. It will reinforce the sarcolemma and help transmit the tension generated by the sarcomeres to the tendons. So, a brief summary of your proteins.